Hello everybody and welcome to today's event. We're just going to give it a couple of minutes just to allow a bit of time for everybody to join. There are going to be a couple of poll questions that should pop up on your teams in the next minute or so. Just 2 questions gives us a bit more of an idea about your area of practice and just some sort of baseline knowledge in genomics. So if you're happy to complete those, it'd be really helpful for us. It just helps inform our education going forward. So I'll give you a minute to complete those and then we'll get started.
No.
Right, I think we're a couple of minutes.
Sorry, that's it. Thank you, Sandra.
Just a couple of minutes past now, so we'll get started.
Thank
Just some
you.
quick
Sorry about that. Always a drama with me. Thank you.
Before we start, could we just ask that everybody please mute their microphones for the duration of this session. There will be time for questions at the end, and there's also a chat function if you have questions. So please, if you've got questions at the end, unmute and ask. This session will be recorded and the recording will be shared afterwards. If we go to the next slide, please, Salim. So a very warm welcome to everybody who has joined us in today's session, which is the first in a series of national genomics lunch and learns. Today's session, we're going to be talking about what genomics is and why it's relevant to you as healthcare professionals, no matter what your area of practice. Next slide, please. My name is Marianne Quinn. I am the lead genomics midwife at the South East Genomic Medicine Service. I'm joined today by Glenda. Glenda, I will hand over to you to introduce yourself.
Hi, I'm Glenda Beaman. I'm the Education, Training and Workforce Lead for the Northwest GMSA. Welcome.
Next slide, please. And again. So again, a very warm welcome, and it's so exciting to see so many of you here on the call with us today. So over the next 45 minutes, we're going to be talking about what genomics is and more importantly, why it's important that all healthcare professionals have a good understanding of the fundamental principles of genomics. So we're going to be sort of covering those fundamental principles, including chromosomes, genes and inheritance, with some real life case studies taken from practice. We're also going to introduce you to the concept of epigenetics, and then Glenda's going to talk through some of the many educational resources that we have available, as well as the Genomic Medicine Service and the role that plays in supporting you. And as I said, this is the first in a series of monthly national genomic lunch and learn. So we're going to introduce you to the series as a whole and what we're offering and hoping to achieve with that series. Right, next slide, please. So before we start, I think it's a really important point to make is that genomics is not the future of healthcare. It's happening in healthcare right now. So there are many specialties, including cardiac nurses, cancer nurses, screening and fetal medicine midwives that are using genomics every day, and genomics is very much embedded in their practice. But as we understand more and more about genomics, its scope is much greater, can now be involved in disease diagnoses and personalised treatment and ultimately in prevention. And therefore, it's really important that all healthcare professionals have a good understanding of genomics and how it applies to their practice. And as I said, there are many educational resources that are available to support you in this, and we will talk you through some of those today. Next slide. So the first question that was the title of today's session is, what is genomics? And very helpfully, the World Health Organization have actually created a video that answers that very question. It's just 90 seconds long, and we will play that for you now.
Think of the world like a library. Every one of us is a unique book filled with pages that describe why we are the way we are. The color of our eyes, the lobes of our ears, the size of our smile, the beat of our heart. It's a book that's written in its own language, a genetic code of A's, C's, G's, and T's. It holds the story of our families, our ancestors, our lineage surging through our veins. The study of these pages, that's genomics. Genomics is how we understand what makes us so similar, yet so unique. It's how we make sense of our DNA, the instructions for how our bodies function. Genomics has the power to create medicines and prevent diseases. It's how we can use our diversity to improve our future. The story of genomics is the story of our lives. And where that story leads is up to us.
Next slide, please. So as you saw in that video, I think it's a really, really lovely video that explains genomics really well. A few technical issues at the beginning there, but it wouldn't be a Teams call without those. But I think hopefully most of you managed to catch the message there for the video. So to put simply, what is genomics? Well, genomics is the story of us. And why is that relevant to you as healthcare professionals? What are you understanding this story, the code that makes us who we are, that tells our bodies how to function? Can we understand why some people are predisposed to certain disorders and how these disorders run in families? It explains why if two people get the same disease, they can suffer very different symptoms. So some can have very few symptoms and others can end up critically ill with the same disease. And COVID was a prime example of this. And then we can use this knowledge and use that into how do we treat disease. And again, it answers some of these questions about why when we give a treatment, a medication, do some people respond very well to that and other people have very adverse reactions to that medication. And then we can use all of this information together and ultimately what we all want to achieve, which is disease prevention and better outcomes for people. So if you take only one message home today, why is it relevant? It's because it is us. Genomics is all about us. It's understanding who we are and how we function. But there is no denying that genomics is a complex subject. So a popular analogy that's often used when we're explaining genomics is, as that video says, that the world is a library and we are all unique books. So our books are made up of many, many letters, and these are our nucleotides. These pair to form our DNA. We have over 3.2 billion letters in our story. These letters are then arranged into sentences, and a sentence that codes for a protein is known as a gene, and we have 20 to 30,000 genes in our story. All of these sentences and letters are organised into chapters, just as all of our nucleotides or our DNA and our genes are organised into our chromosomes. So we have 23 pairs of chromosomes, or there are 46 chapters to our story. And if you were to read that cover to cover, that's your genome. So it is everything that makes you who you are, and no two are the same. And to put that into some sort of perspective, because these are big numbers and a lot of information, if you were to read your genome cover to cover at a sort of leisurely sort of rate of one base or nucleotide per second, it would take you about 100 years to complete that story. So it is a huge amount of information. Very luckily, we have technology now that can do that in about a day. Next slide, please. And that sort of vast amount of information is stored in nearly all of our cells. So if we look at a cell here, within the nucleus are all of our chromosomes, which, as I said, are those chapters which store all of our genes and our nucleotides. If we zoom in on one of those chromosomes, we will see that it is made up of many millions of bases or letters which base pair to form our DNA, and a sort of segment of that DNA forms a gene. So a gene codes for a protein, and a protein has a molecular impact on our bodies. Next slide, please. I think a comment that's often made is, what is the difference between genetics and genomics? Because I've heard of genetics, but what is the difference between genetics and genomics? And historically, we very much focused on genetics. So genetics is the study of our genes. As I said, we have 20 to 30,000 genes in our genome, and genes code for a protein, and proteins tell our bodies how to function. And we inherit our genes from our parents. So we have 2 copies of all of our genes. We get 1 from each biological parent. And the thought is that if there are changes in the disease in a gene which stop the protein from functioning, then this can cause disease. And since these are inherited, run in families, we should be looking at our genes to understand how we get disease. And this still holds true, but our genes are only a small amount of our genome, so they only account for about 2% of our genome. And even though they're a very, very important part of our story, they don't tell us everything. So our genome is looking at the whole story of us from cover to cover. So that is every single letter, those 3.2 billion letters that make us who we are. So quite remarkably, if we think about our genome and we compare that to the genome of other species, we are remarkably alike. So if we look at a chimp, we're 96% identical. If we look at a mouse, 90%, we're 60% identical to a banana. If I were to read the genome of each and every person that was on this call today, we would find that we were all 99.9% identical. So we use that analogy of a story. All those letters are 99.9% the same. So again, the question is, why are we concerned about genomics if we're all so alike? And that's because that 0.1% accounts for up to 3 million variations, so 3 million differences to our stories. And the power of genomics is really trying to understand what do these differences mean. So most of these differences will be normal and they don't have any impact on our health. These are normal, healthy variations. So again, if we were thinking about our genome as a story, if I was reading a story and the beginning of a sentence didn't have a capital letter, this is a change or a mistake, but it doesn't impact how I understand that sentence or how I understand that story. However, if we start removing all the vowels, if we start removing sentences altogether or removing whole chapters, that's going to have a much larger impact. And the power of genomics is trying to understand variation and then apply this to personalize disease diagnoses, treatment, and ultimately to prevention. The next slide, please. And so as we discussed at the very beginning, when we talk about our chromosomes, and this is where all of those bases or nucleotides and genes are stored, we have 23 pairs of chromosomes in each of our cells. The only cells that don't have 23 pairs are the egg and the sperm, which contain half the amount. And when fertilization happens, it brings that number to a whole genome. And why is it important, though, that we understand? Why does it matter how this genomic information is stored? And that's because we know that there can be errors in meiosis which change the number of chromosomes that we have. And this numerical chromosome change is called an aneuploidy. So just the picture here shows a karyotype that's all very neatly organized chromosomes. So you've got numbers 1 through to 22, and we all have those. The only difference are the sex chromosomes, which are XX in a female and XY in a male. Next slide, please. So I would say one of the most well-known numerical chromosome changes is Down syndrome. A person with Down syndrome has got an additional copy of chromosome 21. So we have 2 copies. A person with Down syndrome has an additional copy of chromosome 21. And we can use our sort of genomic understanding to understand why Down syndrome is the most well-known chromosome numerical disorder. And that's because we all know somebody with Down syndrome. We see people with Down syndrome in society. The phenotype of people with Down syndrome can be very variable. But for the most part, they live very fulfilling and semi-independent lives. And that's because chromosome 21 is the smallest chromosome out of all the chromosomes that we have, so it counts for only 1.4% of the genome. So if you have an additional copy of that chromosome 21, it has less of an impact than if you say had an additional copy of chromosome 1 that you can see is much larger. And if you had a trisomy 1, this would actually be universally fatal. So we can use the sort of understanding of genomics and how our information is stored and apply this into how a disorder is likely to present. Next slide, please. But when we're thinking about genetics and genomics, we often do look at our genes. So as I said, our genes are a sort of a stretch of letters that code for a protein, and proteins tell our body how to function. And as I said previously, we have 2 copies of all of our 20 to 30,000 genes. We inherit 1 from each biological parent. And by understanding if there are changes in these genes, how that's going to present, whether somebody gets a disorder or not, we have to look at inheritance patterns. And the one displayed here is a dominant disorder. So if we look at the diagram there, we've got somebody who has got a change in 1 of the copies of their genes, and they have the disorder. If they had a child with someone who does not have a change in either copy of the genes, they've got 2 normal copies, there is a 50% chance that each of their children, each time they become pregnant, the child will inherit that change from one of their parents and have the disorder like their parent does. And there's also a 50% chance that they will be unaffected. And some of the most well-known dominant disorders are Huntington's disease, osteogenesis imperfecta, and Lynch syndrome. And if you look at the family tree here, so the squares are males and the circles are females, you'll see that this will present in multiple generations, and it doesn't it will affect males and females equally. We go to the next slide, please. So again, one of the key questions is, you can talk about dominance, so a change in 1 copy of the gene which causes the disease, but why does this matter to my practice? And that's because we know that if we can identify that a disease is autosomal dominant, then this can have really far-reaching implications, not only for that individual person, but for the wider family. So if we look at Andrea's case example, which was taken from clinical practice, so Andrea's mum was diagnosed with bowel cancer. So we know that bowel cancer is associated with Lynch syndrome. So Lynch syndrome is a dominant disorder, so caused by a change in a single gene copy. And if you have Lynch syndrome, it greatly increases your lifetime risk of bowel and endometrial cancer. So Andrea's mum had genetic testing, which confirmed that she did have one of these changes. So she did have Lynch syndrome, which was likely cause of her bowel cancer. So Andrea herself, she was young, she's fit and well, but she chose to have genetic predictive testing because she knew that as her mum had this change, there was a 50% chance that she will have inherited that changed copy and will also have Lynch syndrome. Even at the moment, she has absolutely no symptoms of Lynch syndrome. Andrea has this testing and it confirms that she has inherited this change, and therefore she has an increased lifetime risk of bowel and endometrial cancer. And again, why does that matter? And that's because, but now that we know this about Andrea, we can enter her into comprehensive screening for bowel cancer, so that if she were to get cancer, we're going to detect this much earlier and she's going to have a better outcome, hopefully. She can choose to have risk-reducing surgery, and she can access support, meet other people who also have Lynch syndrome, so can form that sort of community and psychological support. And then this has even further reaching implications, because if she goes on to have children of her own, she now has access to reproductive options, which can mean that embryos are selected that don't have this gene change, so she doesn't pass on this change to her children. So her mum's diagnosis has had a huge impact on what we are able to do for Andrea, because we understand how this gene change is inherited and what impact that's likely to have on health. Now, next slide, please. So autosomal recessive disorders, in this case, you need both copies of that gene to have the change in order to have the disorder. So unlike in dominant, where you only need 1 copy to change, this one you need both copies to change. So if we look at the top picture there, if you have only 1 copy of the gene that has changed, you will be a carrier of that change. The vast majority of carriers are completely asymptomatic, so they have actually no symptoms, they're fit and well, and they wouldn't have a clue that they were a carrier unless they went to have genetic testing. The reason it's important to understand this is because if a carrier of a gene change has a baby with a person who also has a gene change in that gene, there is a 25% chance that their children will inherit both copies of the changed gene and have the disorder. So you need, if you look at the bottom left here, you need to inherit both changes, that both copies of the gene have the change to get the disorder. And well-known examples of recessive disorders are cystic fibrosis, sickle cell disease, and thalassemia. Again, this will present very differently. So unlike in dominant, where you would see it through many, many generations, often there's no family history at all of the disorder, and people only know they're a carrier when they have a child that is affected with the condition. Next slide, please. But again, why is it important that each and every one of us is aware of what recessive disorders are and how they're likely to present and how they're inherited? And we could use Lucy's story as a key example of this. So Lucy is 8 weeks pregnant with her second child. First child is fit and well. Lucy's sister has just had a baby who was diagnosed with cystic fibrosis at birth on the newborn blood spot, which is offered to all babies at 5 days old. Other than that, there's no other family history of cystic fibrosis within the family. The community midwife that was booking Lucy's pregnancy realised that because Lucy's sister has had a baby with cystic fibrosis, Lucy's sister must be a carrier of a change in the cystic fibrosis gene, and therefore Lucy's parents are like one of them, at the least, was likely to be a carrier of this change. So Lucy herself is at increased chance that she has inherited a change in the cystic fibrosis gene. So this community midwife referred Lucy in to the screening team, and they were able to screen for variants or changes in the cystic fibrosis gene for Lucy and her partner. This testing identified that both Lucy and her partner had a change in the cystic fibrosis gene. So you would think that perhaps this is very bad luck, but actually being a carrier of a cystic fibrosis change is quite common, and 1 in 25 Caucasians are a carrier of this change and wouldn't know about it unless they had this testing. So because we identified that both Lucy and her partner have got this change in the cystic fibrosis gene, so they themselves are completely well, but there is a 1 in 4 chance that baby will inherit both copies of the changed gene and have cystic fibrosis. Lucy was offered diagnostic testing at 11 weeks, which confirmed that the baby did not inherit this changed gene, but had inherited both normal copies from parents. So this had a big impact on her pregnancy. We were able to provide reassurance at an early gestation, but it also has far-reaching implications for all future pregnancies. So we can now offer genomic testing to Lucy from as early as 8 weeks via a non-invasive prenatal diagnosis. So a simple blood test from mum's arm can determine whether that baby in subsequent pregnancies has got cystic fibrosis, and we'll talk more about this later on in the series. But again, it just highlights how the diagnosis in a family member has had real significant impact on Lucy's pregnancy journey. Next slide, please. So finally, I'm just going to touch on X-linked disorders, which is another form of inheritance. It kind of combines dominant, where you need 1 copy, and recessive, where you need the 2. So if we think back when I was talking about chromosomes, we have those 23 pairs of chromosomes, so numbers 1 to 22, and then our sex chromosomes. So a female has got 2 X chromosomes, whereas a male has got 1 X and 1 Y. So with X-linked disorders, if you have a gene change on the X chromosome, if this occurred in the male, he doesn't have another X chromosome. So this gene change will cause the disease. If he goes on to have children, all of his daughters, because daughters will always get an X from their father and an X from their mother, will be carriers. So sometimes unknown carriers of this disorder. If these carriers then go on to have children, so the reason they don't have the disorder is because they've got that extra X chromosome where the gene is unchanged. But if they go on to have children and that child is a male, there is a 50% chance they will inherit the X chromosome that has got that gene change and will have the disorder. So well-known examples of this are Duchenne muscular dystrophy, Fragile X syndrome, and haemophilia. And often, I mean, there are many, many case studies of Duchenne muscular dystrophy online where mothers are complete unaware that they're carriers, and it's only when their child is diagnosed with muscular dystrophy at age 2 or 3 that they are aware that they were carriers. And these conditions have really quite devastating impacts on the person and the family. Slide, please. So again, this just highlights what this would look like. If we were asking somebody about their family, we would see that only males, who are the squares, will have the condition, and females will be carriers but will not have any symptoms of the condition. Next slide, please. And another case study which really highlights the benefits here of genomics and how this can be applied in practice is that of Yusuf. So Yusuf was diagnosed with Hemophilia B. So this is an X-linked disorder, so it's due to a change of the X chromosome. And this gene produces a clotting factor, which helps our blood clot. So because he's got this change in the gene, his blood doesn't clot as it should. He lived in Egypt as a child, so couldn't access medication that he needed for his haemophilia. So his parents sent him to the UK, where he was able to get the treatment that he needed. But a research nurse then approached him and spoke to him about the possibility of having gene therapy. So therapies are now available where we can introduce a normal, healthy copy of the gene to replace the one that is not functioning, is not able to produce the protein that our bodies need. And in 2021, he was the first adult to have gene therapy for haemophilia. And it's just a quote from Yusuf, who says, "I can now do the things I couldn't do before, like playing football and riding my bike." So these things that most of us take for granted, the power of genomics is that he's now able to do these things. So it's, I think, a really powerful message about what we can do with this information and how it can influence the treatments that we can offer. Next slide, please. I think there's no question that sometimes communicating this with our patients can feel very overwhelming and complicated, but there is a fantastic resource called GeNotes, which has created many visual guides that you can share with your patients if you're wanting to discuss this. There's also a Knowledge Hub, which actually talks you through almost all of the aspects of genomics, the terminology, and there's an in the clinic option that actually brings you through a lot of in the clinic scenarios. So again, really helpful in applying genomic knowledge into clinical practice. So I would urge you all to please have a look at GeNotes, because it's such a valuable tool in aiding our understanding, and it's got a lot of resources again that you can share with your patients. Next slide, please. So just the last thing that I'm going to touch on is epigenetics, which is, again, undeniably a complex concept to get your heads around. But if we thought about our genome, so the genome is each and every letter that makes us who we are, epigenetics is sort of taking it one step further. So we know that we have 20 to 30,000 genes, but we don't use all of those genes. So some of these genes, like a light switch, are turned off and some are turned on, and this is a normal process, it's a healthy process. So epigenetics looks at how our environment can influence which genes are turned on and which genes are turned off, and how this can be linked to disease. So a popular analogy that's used to explain this is that if you were playing the violin and you gave the violinist a piece of music, an identical piece of music, and you told them to play it in the Royal Albert Hall and on the London Underground, although that piece of music is the same, just as in epigenetics, our DNA code is the exact same, how that music sounds is going to be very different, because environment matters, and it's the same for us, our environment matters. Next slide, please. So this is very much an emerging field of genomics, and not one that you need to know in a huge amount of detail. It's just to be aware of that. And there is a lot of research that's happening at the moment that shows, as this picture shows, many environmental factors can influence which genes we turn on and which genes we turn off. And this then can directly impact not just our physical health, but also our genomic health. So it plays in really to that early intervention agenda and public health agenda, that message of not only showing how things can influence our physical health, but also how these can change our genomic health, and whether there are any treatments and personalised treatments we can look at here. Next slide, please. So just a sort of quick recap on what we've covered so far. What is genomics? I would say genomics is us. It is the story of us. It is what makes us who we are and tells our bodies how to function. And why is it relevant to you? Is because by understanding that story, and don't get me wrong, there is still much that we want to understand, but by understanding that story, we can apply that to disease diagnoses, to disease treatment, and ultimately to disease prevention. So we've very briefly gone over the fundamental principles of genomics and genetics, what chromosomes are, our genes, and how these can be inherited. And again, just touched on epigenetics. Next slide, please.
Okay, thank you. Thank you very much. So I'm going to carry on now with looking at what sort of educational resources are out there to actually support your understanding around genetics and genomics. Next slide, please, Salim. So really what we think about is, because genomics is rapidly becoming a cornerstone of modern healthcare, the nursing and midwifery is no exception. So we expect it to be part about 50% of all healthcare interactions by 2035. From understanding inherited conditions to guiding personalised treatment plans, genomics is influencing how care is delivered across all specialities. Nurses and midwives, often the first point of contact for patients and their families, play a critical role in recognising what genetic or genomic information may impact their care. Whether taking a comprehensive family history, explaining test results, or helping patients navigate complex treatment options, all healthcare professionals are increasingly expected to integrate genomic knowledge into their everyday practice. Next slide, please. So there are currently quite a wealth of educational resources out there to actually support you, whether it's drawing a family history, understanding the basics. A lot of these resources are held within the National Genomics Education Programme, and obviously all the links are actually on each one of these slides for you. And really this is to support the integration of genomics into NHS practice. It offers a range of educational resources for healthcare professionals, and it focuses on building genomic competencies across the workforce. One of the key ones, if you really want to start at a very basic level, and I know we've had some explanation about what DNA is and genes, etc., but you can actually do these little bite-sized educational tools, and they're called Genomics 101, and there's probably 6 or 7 that I would potentially have a go at to give you the really basic understanding and put you back at that level that you may want to be at. And this will really set you in that really true baseline of understanding what's really required moving forward within your profession itself. Next slide, please. Because there's been such a need for understanding genomics around the nursing and midwifery arena, there's been a group of people, including Genomics Education Programme, that have actually developed what they're calling a genomic passport. And that really isn't to frighten anybody, and this isn't, mandatory training or anything. These are tailored roadmaps to help you learn the genomic or understand the genomic competencies. And they're actually broken down into different tiers. So they will break down into very novice levels, intermediate, and expertise. So it really depends on where you want your level to stop. And I think once you get into it, you'll probably want to know more and more and more. So the 2 links that are there, one is for the nurses and the other one is for the midwifery one. They're not too dissimilar, but there are areas on there that will navigate you to respective places according to your professional. So just a quick statement, understanding genomics and its use in healthcare is becoming more relevant to nursing practice, as reflected in its inclusion in the Nursing and Midwifery Council standards of proficiency for registered nurses and midwives. It's important that nurses and midwives are able to talk about genomics to patients and colleagues with confidence, but for many, there is still a need to upskill. And I know a lot of the words that we use within the genomic arena can be quite challenging. So it's better if you undertook a lot of these basic courses, and that will actually make you a little bit more confident and more fluent in being able to explain these to your patients. Next slide, please. So if you feel after you've taken some of the basic courses that are available to you, you can progress, and there is a lot of different avenues that you can go on. And being part of the healthcare professional organisation, you can undertake either an MSc in the whole of genomic medicine, or you can take certain modules within it as well. Now, that is actually funded by NHSE or the NHS. So you can actually select, and all you need to do is to register and fill in the application form, and then you can look at what you want to do from it. So I think it's something, if you really find interesting, you can actually progress your career within that genomic arena. There's also a lot of massive open online courses that sit in FutureLearn. These are all free courses. There's no cost to anything that we're actually highlighting to you. And again, that will tell, there's courses that there's understanding variant interpretations. So if you're talking to a family and they've got a specific change within a gene, you can actually explain what that variant means, whether it's pathogenic, for example, if it's a benign variant or a variant of uncertain significance. Actually understanding and being able to discuss that with your patient really is quite important for you and for your patients as well. Next slide, please. Just if people aren't aware, just to highlight the fact that there are 7 genomic services around the UK, and each one of those is highlighted, and each QR code that's on here will take you to your respective GMSA or GLH. And within each one of those, there will be areas within those that will highlight all the training or resources or support networks that you actually might need. So this is a good area just to sort of get yourself familiar with and look at what's actually being developed within your local area as well. Next slide, please. So just another one that I really wanted to highlight is the Macmillan Genomics toolkit. Now, obviously, this is all cancer related, but they've developed toolkits around breast, colorectal, gynae, lung, prostate, and there are more to follow. But within that, there is a lot of genomics overview, there's lots of training resources, there's lots of webinars, lectures, workshops, etc. But again, it's not just for you, there is areas in there for the patients as well. So some of the resources may be replicated, or they might redirect you to Genomics Education Programme, the Genomics 101, and everything else that's aligned. So somewhere you will pick up a link to direct you to all the education that's actually out there. Next slide, please. And just as an example, this is one that was undertaken by North Eastern Yorkshire and the North West GMSA. This was applying genomics in practice under the National Lung Cancer Initiative. And this was developed by the lead nurses. And again, this was looking at how, what education resources were needed for nurses, specifically along that cancer pathway. And again, if you look on the diagram of the lung, there are 4 little QR codes. So there's Genomic Novice, that would be your Genomics 101. Then you've got the competent, the proficient, and then the expert. And all of the links to education will be graded up as to what level you actually need to actually undertake. Next slide, please. So following up from this, the continuous learning, the upcoming National Genomics Lunch and Learn sessions, these are going to be monthly webinars covering various genomic topics. That'll be ethics, consent, taking a family history, focusing on genomics throughout the lifespan, from antenatal to the end of life care, applying genomics to practice through genomic red flags, designed for flexible and accessible learning, opportunities for questions and answers and discussions, and a schedule and registration details will be available through GMSA platforms. Next slide, please. Just to finally recap. So everything that we've presented today, and I know we've only sort of skimmed the surface, but this is just to highlight things that are available and what things are. So Genomic tells the stories of our lives and is relevant in all areas of clinical practice, from disease diagnosis, treatment and prevention. The fundamental principles of genomics, including chromosomes, genes, inheritance, epigenetics, and application to clinical practice. Genomics Education Programme website, the regional sites for localised support, the Macmillan Genomics Toolkit, GeNotes for point of care information, and the National Genomic Webinar Series. The next slide, please. So as part of what we're doing, it is really, for us, it's really important that we get feedback. So this QR code here, if I could please ask everybody to scan it, if they can. It will only take a minute to do, but this really will help inform what the topics are, what else we might need to put in there, and also might identify any new resources that we might need to develop. And after completion of everything, a certificate of attendance is available for this session, and I'd like to thank everybody in advance. So we'll just leave the QR code on just for a couple of minutes if everybody would like to scan this. And thank you.
We just go on to the next slide just before people leave, because this is the sign up to the next, and then we'll have time for questions. This is the sign up for the next webinar next month. So please do scan and sign up for next month, which will be taking you through the consent and ethics, which is a huge part of genomics. And we then have a session on taking a family history, and then we will take you on a journey through the life, really applying these genomic principles across the lifespan. But please, yes, I think we have potentially a few questions or hands raised. So if you've got any questions, there's also just a quick poll at the end. So please, if you have a second, just complete that for us. And yeah, we'll share that link now for you for the next session. And we can also share the link to this survey as well for you to complete if you have time.
Marianne, can you see if there's any hands up at all? I can't see on mine.
Don't think there are, from what I can see. But if anyone's got any questions, please do ask. Oh yeah.
Effects, I don't know who
Tina. Florentina.
Hi, yeah, it's Flori. I work in Sheffield in genetics.
Hi.
Hi, thank you for this presentation, really nice. Wanted to highlight the non-coding protein genes. I know you gave a, I saw you gave a beautiful description about genetics and genomics, and the fact that most genes lead to a protein. However, there are some genes that don't lead to a protein, but they are still very important and have unique functions within the genome. So yeah, I don't know if that's something you could maybe incorporate in the future and create awareness about it.
Thank you, that's really helpful. And Absolutely. I think today we covered the fundamentals, but I'll make sure that we sort of incorporate that and raise awareness, because you're right. Really, this is something that we're learning more and more about, that we're more than just the sort of proteins, the genes and the proteins that they code for. Some genes don't code for a protein, but they still have an impact, and even all those non-coding regions. But yeah, thank you for that comment. That's really, really helpful. We'll incorporate that going forward.
Yeah, Salim, could you put the QR code back up for feedback, please? Go back one slide. Thank you.